pizza, cupcake, pasta, ice cream, milkshake. Well, this is not a food fest. These are just clues for y'all to figure out what the topic of this session is. Any guesses? Well, yes, you're right. It's about carbohydrates. So the topic of the session is chemistry of carbohydrates. So to briefly tell you what are carbohydrates, carbohydrates are quite important for our body. They have multiple functions. They are defined as polyhydroxyaldehyde or ketone. Poly means many. Hydroxy is a hydroxy group which is present. So there are many hydroxy groups in the carbohydrate. In addition to that, there may be an aldehyde group or a ketone group. So aldehyde group is often referred to as CHO and keto is a C double bond O. So these are the functional groups usually present in carbohydrates. Or any compounds which can yield these polyhydroxyaldehydes or ketones can also be called as carbohydrates. So the general formula is usually CnH2ON. So from the formula itself, you can make out that the three main atoms that are present in carbohydrates are carbon, hydrogen, and oxygen. Now, depending on the number of carbon atoms present in the carbohydrate, you can arrive at the molecular formula of a carbohydrate. Say, for example, a carbohydrate has 6 carbon atoms. So if you substitute in the formula that is given here, you get it something like C6H2O, the whole 6. Or in other words, we can write this as C6H12O6. So this is the molecular formula of a carbohydrate which has 6 carbon atoms. Now, carbohydrates are also called as saccharides. Saccharides is a Greek word which means sugar. So if I speak anything about saccharide, it literally means carbohydrates. Coming to the function of carbohydrates, there are so many functions in our body. I've just listed some of the main or the major functions of carbohydrates. The first and the most important one is energy source. Now, carbohydrates are known to give us energy, and especially glucose, which is utilized by most cells in our body, are utilized basically for energy purpose. Then, carbohydrates can also be stored for energy purpose that can be utilized later on. So we have examples like starch and glycogen. Starch is the storage form of carbohydrates in plants, whereas glycogen is the storage form of carbohydrates in animals. Next, carbohydrates can form structural components. Carbohydrates are typically present in the cell membrane or the membrane that envelopes a cell. So usually carbohydrates are present in the form of glycoproteins or glycolipids, where carbohydrates are complexed with proteins and lipids in the cell membrane. Carbohydrates can also play a role in the formation of cell receptors and also various other structures, especially when it makes up the ground substances of various tissues like the cartilage, bone, cornea, lens, etc. Carbohydrates can also act as a precursor. Glucose, which is a carbohydrate, can form a lot of substances in our body. So like that, you have several other carbohydrates that can act as precursors for various other physiologically important compounds. Then they also are dietary fibers. We have certain examples like cellulose, hemicellulose, pectin. All these are examples of carbohydrates which can act as dietary fibers. They are present in leafy vegetables and the fruits. So by taking in dietary fibers, it helps us prevent constipation, and it also is known to protect us from colon carcinoma. So this is another function of carbohydrates. The last one is carbohydrates are important constituents of nucleic acids such as DNA and RNA. Now, as you know, nucleic acids or DNA or RNA is made of a pentose sugar, a phosphate group, and a nitrogenous base. So the pentose sugar is a carbohydrate again, and then in DNA you have a deoxyribose, which is the pentose sugar, whereas in RNA you have a ribose sugar, which is again a pentose sugar. So this is in brief about the functions of carbohydrate. Additionally, there are other functions as well. Certain blood group substances can also contain carbohydrates like mannose, glucose, etc. And you have certain drugs known as glycosides, which are examples of carbohydrates. Again, I can give you the example of digoxin, which is used in the treatment of cardiac failure. So this is an example of a carbohydrate used in treatment. Now coming to the classification of carbohydrate, in general, carbohydrates are classified based on the number of sugar units or the saccharide units. So we have monosaccharides, disaccharides, oligosaccharides and polysaccharides. Now what do you mean by this? Mono means one. So monosaccharides are known to have one sugar unit. The word di in disaccharide means two, hence disaccharides have two sugar units. Then we have oligosaccharide. Oligo means few. So in oligosaccharides there are around 3 to 10 sugar units. And then finally we have polysaccharides. Poly means many. So we have more than 10 sugar units in polysaccharides. So this is in brief how carbohydrates as a whole is classified based on the number of sugar units. Now moving on to monosaccharide, which is the most simplest form of the carbohydrate because there's only one sugar unit. So it cannot be broken down any more further. So this is simplest carbohydrate, cannot be hydrolyzed further, and they are known as reducing sugars. All the monosaccharides known are reducing sugars. What do you mean by reducing sugar? So reducing sugar is a sugar which contains a free aldehyde or a free keto group that is capable of undergoing a reaction to either probably form a glycosidic bond, that is a bond between a carbohydrate and a carbohydrate, or carbohydrate and a non-carbohydrate, or it could these aldehyde or the keto group is capable of reducing other substances. So that's the meaning of reducing sugars. There are free aldehyde or free keto groups present in the carbohydrate. Now again, monosaccharides are classified further based on two things. One is the number of carbon atoms and the second is the type of functional group present. So as I've said earlier during the definition, there are two functional groups that may be present in a carbohydrate. You may have an aldehyde group, CHO group, or you may have a keto group, C double bond O. So now let's have a look at how monosaccharides are further classified based on these two factors. So based on the number of carbon atoms, we have different types of classification. We have triosis, which means there are 3 carbon atoms. We have tetrosis, which means 4 carbon atoms. Pentosis, which means 5. Hexosis, which means 6 carbon atoms. And then lastly, we have heptosis, which means 7 carbon atoms are present in the monosaccharide. Now, this is one way you can classify monosaccharides. On the other hand, based on the functional group present, whether there's an aldehyde group or a keto group, you have two subclassifications of monosaccharide. So you have aldoses and ketosis. So from the name itself, you can figure out what is the functional group present. In aldoses, the functional group is an aldehyde group, whereas in ketosis, the functional group is a keto group. And usually, the aldehyde group will be first carbon in the carbohydrate. Whereas in ketosis, the keto group is usually present at the second carbon atom of the carbohydrate. So this is it about the classification of monosaccharides based on functional group. Now combining those two classifications, that is based on number of carbon atoms as well as the type of functional group present, I have put up a table over here that shows you examples of different carbohydrates under those two classifications. So I've listed out the number of carbon atoms here, starting with triosis, tetrosis, pentosis, hexosis and heptosis, and then the corresponding molecular formula has been calculated and put up here based on the general formula CnH2ON. Now, well, let's have a look at what are the different examples of an aldose sugar and ketose sugar depending on the number of carbon atoms present in the carbohydrate. So example for triosis, the aldose sugar is called as glyceraldehyde, whereas the corresponding ketose sugar is known as dihydroxyacetone. Moving on to tetrosis, which contain 4 carbon atoms, the aldose sugar is known as erythrose, whereas the corresponding ketose sugar is known as erythrulose. Coming to pentosis, that is carbohydrates which contain 5 carbon atoms, the aldose sugar is ribose and the corresponding keto sugar is ribulose. Likewise, we also have another one which is not mentioned here. Xylose is an example of a pentose sugar which is an aldose sugar, and then you have the corresponding keto sugar which is xylulose. So these are the examples of pentoses. Moving on to hexoses, or those carbohydrates which contain 6 carbon atoms. The aldose sugar examples are glucose, galactose and mannose, whereas the corresponding keto sugar is fructose. And then the last one is heptoses, that is carbohydrates or monosaccharides which contain 7 carbon atoms. The aldose sugar is glucoheptose, whereas the corresponding keto sugar is pseudoheptulose. So this is in brief about the examples of various monosaccharides based on the number of carbon atoms as well as the functional group. Now, just to give you a fair idea of what is the difference between those two, the aldose sugar and the ketose sugar, I've just put up the structures over here. So generally, in order to number the carbon atoms, you start off with the first carbon atom that is on top. So this will become the first carbon atom in a monosaccharide, and then this will become the second, and then this is the third. So usually the terminal carbon will have a hydroxy, an alcohol group like this, and it's always written like this. So glyceraldehyde is the most simplest form of carbohydrates. As you can see, there are 3 carbon atoms, and it's an aldose sugar because it contains a functional group which is CHO. And in case of the corresponding ketose sugar, which is dihydroxyacetone, the functional group will be at C2. And this is how it is written. The structural formula is written like this. Next, the same thing with erythrose and erythrulose. You can see there are 4 carbon atoms. You start numbering from top. So here again, the functional group aldehyde is present at the first carbon atom, and the keto group is present at the second carbon atom. The next one is ribose and ribulose. Again, there are 5 carbon atoms, and you can see how the functional group is present in both these two. That's how you're able to differentiate between an aldose sugar and a ketose sugar based on the functional group. Then we have glucose, which is a heptose, aldoheptose, and is an aldose sugar. Again, there are 6 carbon atoms in both, and you can see fructose, which is a ketohexose. The only difference over here is the presence of the functional group, that is aldehyde in case of glucose and keto group present in fructose. And the last one, you can have a look. This is an example. These two are examples of heptoses, that is 7 carbon atoms are present. Again, everything is the same except with in respect to the functional group. In these two, you have an aldehyde group in aldoheptose, whereas a keto group in pseudoheptulose. So this was in brief about monosaccharides. Now we'll move on to the next one, which are disaccharides. As the name suggests, there are 2 sugar units or 2 saccharide units, and these 2 sugar units are joined together. Imagine this is one sugar unit and this is the second sugar unit, or a monosaccharide. Okay, one monosaccharide and the other monosaccharide, and there will be a bond in between. This bond joining the 2 sugar units is known as glycosidic bond. So there are 2 monosaccharide units joined together by a glycosidic bond. Again, disaccharides can also be classified based on whether they are reducing or they are non-reducing. So under reducing disaccharides, we have 2 examples, that is maltose and lactose. Maltose is also known as malt sugar, and it consists of 2 monosaccharide units joined together by a glycosidic bond. The 2 sugar units are glucose and glucose. Then we have lactose, which is present in milk. Again, you have 2 sugar units or 2 monosaccharide units, glucose and galactose joined together by a glycosidic bond. These are reducing disaccharides. On the other hand, we have non-reducing disaccharides. An example of that is sucrose. Sucrose is present in your common sugar, table sugar that we use in food. So sucrose is again made of 2 sugar units or 2 monosaccharide units, and there is glucose and fructose joined together by a glycosidic bond. Again, there's another example that I can give you of a non-reducing. It's called as trehalose. trehalose is present in mushrooms. Again here, the two sugar units are glucose and glucose. You may wonder what's the difference then between maltose and trehalose. The only difference between the two is the kind of glycosidic bond formed between glucose and glucose. The attachment of the bonds are at different carbon atoms of glucose and glucose, that is in case of trehalose and maltose. So these are examples of disaccharides. Moving on to the next one, which are oligosaccharides. Oligo means few. So there are about 3 to 10 sugar units or monosaccharide units present, and all of those units, individual units, are joined together by a glycosidic linkage. So you have a bond formed between each of the sugar unit, known as glycosidic linkage. Now the examples of oligosaccharides are maltotriose. As the name suggests, it sounds similar like maltose. So if you remember, maltose had 2 sugar units, maltotriose, tri meaning 3. So there are around 3 glucose units present in maltotriose. Whereas in raffinose, you have a fructose, you have galactose and glucose joined together by a glycosidic linkage. Again, there is stachyose, which contains galactose, 2 molecules of galactose, a glucose and fructose. So this was about the examples of oligosaccharides. Moving on to the last one, polysaccharides. Poly means many. So there are more than 10 monosaccharide units joined together by a glycosidic linkage or glycosidic bond, and they can be further classified into homopolysaccharides and heteropolysaccharide. Homo means same and hetero means different. So if you have a look at the monosaccharide units present in a homopolysaccharide, you'll find the same monosaccharide in the homopolysaccharide. So I'll just give you an instance where there's a homopolysaccharide and the sugar unit is glucose. So you'll have the same repeating glucose unit occurring in the structure. And this goes on. So this is an example of a homopolysaccharide, also known as homoglycans, where the same units are just repeated. On the other hand, in case of heteropolysaccharide, you have different monosaccharide units linked together by a glycosidic linkage. So for instance, I'll give you an example where you have one glucose, then probably you have a galactose, then again a glucose, again a glucose, then a galactose, and then so on. And this keeps repeating, you have different types of monosaccharides present. It's not the same. So this is an instance of a heteropolysaccharide, also known as heteroglycans. So coming to homopolysaccharides or homoglycans, these are polysaccharides which are made of only one type of monosaccharide unit. Again, these units are linked together by a glycosidic linkage. So there are different examples. We have starch, which is the storage form of carbohydrates in plants. It is made of repeating glucose units. Then we have glycogen, which is the storage form of carbohydrates in animals. Again, the repeating unit is glucose. And then cellulose, which is an important dietary fiber. It helps in preventing constipation. Again, is made of glucose. And then there is inulin. Inulin is basically used especially as a renal function test kind of parameter where you can estimate glomerular filtration rate of a patient by measuring the clearance of the substance known as inulin. Again, is a homopolysaccharide which contains repeating units of fructose. So this is about the examples of homopolysaccharides. The next one, which are heteropolysaccharides or heteroglycans. Here you have different types of monosaccharide units linked together by a glycosidic linkage. Examples is that of agar, which is used as a media, especially for cell culture and even in protein electrophoresis, which is a method for separating proteins, serum proteins. And then you have something known as glycosaminoglycans, also known as mucopolysaccharide, which makes up the structural, or you can see the ground substance of various different tissues in our body. So to briefly summarize what was done in this session, carbohydrates are polyhydroxyaldehydes or ketones or any substances which yield those on hydrolysis. Carbohydrates are broadly classified based on the number of sugar units. You have monosaccharides, disaccharides, oligosaccharides and polysaccharides. Monosaccharides are further divided based on the number of carbon atoms and based on the type of functional group as aldose sugar and ketose sugar. Again, disaccharide, which contains two sugar units or monosaccharide units, are classified into reducing and non-reducing. Under reducing, we have maltose and lactose, and non-reducing, we have sucrose. Oligosaccharides, on the other hand, are those made of 3 to 10 monosaccharide units. The examples are maltotriose, raffinose, and stachyose. And finally, I spoke about polysaccharides, which contain more than 10 sugar units. So we have homopolysaccharides and heteropolysaccharides. Homopolysaccharides are those which contain the same sugar units. So examples are starch, glycogen, etc. Whereas heteropolysaccharides are made of different sugar units. So examples are agar and mucopolysaccharides. So that's all for this session.